looking at the vertebrates today, even though last time we also looked at the vertebrates. We were looking at the lamprey as probably a true vertebrate, the hyperarchaea, and then the hagfish, remember, was kind of like a quasi-vertebrate. It had a cranium, which is a vertebrate feature, but they do not have a true vertebrae. So anyway, if we were to look at our classification of vertebrates, we're looking at the phylum Chordata, subphylum Vertebrata, and remember, I artificially put Myxine in there, although if you were to look up the true classification, Myxine really isn't a part of the Vertebrata. It's part of what's called the Craniata, which is somewhere between subphylum and phylum, just for that group. So, probably the organization should be changed, but it's hard to change when it's been so long. Anyway, so last time I mentioned that the class Myxine, the hagfish, and the hyperarchaea, which is the lamprey, were generally classified as what's called agnatha. And so the idea there is that if we were to look at, let's see here.
Oh, okay, but then
Here
there was
is
one class that I was doing something like this, but like we were only doing the last one when I came back, and I was like, that's a lot.
So here we have kind of a side view of the head of a lamprey. And if you remember what they had here, they had, let me use a black pen here for this, they had a cranium. Which was a brain case, a cartilaginous brain case. They have a vertebrae, which is a combination of the notochord and the nerve cord. Okay, and then they had specifically 7 gill arches. 1, 2, 3, 4, 5, 6, 7. So 1, 2, 3, 4, 5, 6, 7 gill arches. Okay, and that's kind of how they were set up. Basically, the gill arches were for respiration and for, in the larval stage anyway, feeding. They use the gill arches for filter feeding. But we have these 7 gill arches suspended off the bottom of the brain. So the question then is, when we go to the next group here, the class Chondrichthyes includes the cartilaginous fishes. Remember, chondrocytes were cartilage cells. So the term ichthyus refers to the fishes in Latin. So the chondrichthyes are cartilaginous fish, and we're looking at today the osteichthyes, which is a bony fish, meaning that their skeleton has been replaced with bone. So these are the 2 classes we're covering today. We'll cover these two till we get back, and these 2 the third till we get back. But this whole group, everything from the chondrichthyes down, are considered to be what are called the Gnathostomata. These are in quotes because they're not true classification names, but they're just a good way to kind of generally describe them. So the question then is, where did the jaws come from? And if we look at, say for example, a shark, so what we have here is, this is the business end of a shark. This is like a souvenir like we used to be able to get in Hawaii. Now this is like forbidden. The sharks are kind of conserved now. But anyway, this is like a souvenir from many years ago. But this is essentially the jaws of I think this is probably some sort of sand shark or something like that. So this is the jaws of the shark, used for chomping and feeding. And if you look more closely, the jaws, this is the jaws from one of the dogfish sharks like we're going to dissect today. The jaws actually are made up of 2 separate arches. There is a mandibular arch in the front, which is this one. And then this one has a mandibular arch, but also what's called a hyoid arch as a secondary arch. And then this is a more complete one. This is a dogfish shark like we're looking at today, but with all the gills and the jaws and the gills and the cranium all together. And what's kind of clear, or what I'm going to try to convince you, is that the jaws actually evolved from the first gill. So check this out. Here is the shark feature. Here's kind of like what the shark has going on. Body into it. This, there's like a shark there. And what they have going on is they have a cranium. And a backbone, the first vertebrae. Okay. And the jaws basically are made up of two separate arch structures that work together. There is what's called the mandible. Mandibular arch. So the mandibular arch is the mandible arch. And then right behind it is a support arch. It's called the hyoid arch. And the cartilage is so flexible, and you're going to see that when you check out the sharks today, that the cartilage you can easily slice through with a scalpel or cut through with scissors. Cartilage is a really flexible, kind of soft material. So when this mandible arch was fresh, when it was taken out of the shark that died, was killed, it was like really, you could actually fold it in half and kind of twist it. And so it's not really a rigid thing. So by itself, this is not a very good chomper. But, and I'll tell you what label A is here in a second. But what you need, or what they need, is a support frame. And this is kind of like a bicycle frame. You know how bicycle frames have kind of like the front triangle and the back triangle, and the two triangles kind of work together to make the bicycle frame for a roof. That's what the shark jaw is. It's a combination of different structures. The mandibular arch is the front arch, which is not labeled here, but the back arch, which is labeled B, is called the hyoid arch. And together, it kind of provides like a frame or like a support arch behind the chomping arch. So these two together are basically what make up the jaws. Is the mandible arch and the hyoid arch combined. So I have the hyoid arch labeled B on this guy here. I have the mandibular arch labeled A on this one. Just so I can keep them separate. So, but you should recognize that the mandibular arch is the front arch on this one. The one with the teeth on it is the mandibular arch. The hyoid arch has no teeth. Speaking of teeth, the teeth, because these are cartilage structures, the teeth are not derived from the arches at all. The teeth are modified scales. If you look at the scales of a shark, and I will encourage you to do this, when you pull the sharks out, use your cell phone and zoom in on the back of the shark. And you'll see that the shark's scales, the back of the shark is covered in these what are called placoid scales, or oftentimes they're called dermal denticles. And dermal denticles literally means skin teeth. And so these are like little teeth on their skin. And then where the skin wraps around the mandible, you can kind of see that on this one, that where the skin had wrapped around the mandible, at the very, very end of the skin is where the teeth are. So the teeth are actually modified scales. So the scales are what form the teeth. And you probably have heard that sharks can shed their teeth, like they're constantly losing teeth and replacing them. That's because the teeth are basically their skin. They're kind of like replacing them like you would like an epithelium,
That's crazy.
Skin. But these teeth are actually not derived from the jaws, they're derived from skin or the scales that are wrapped around the front of the mandibular arch. So that'd be a good test question. That's kind of what this one was I said the structure in A is the mandibular arch, but the letter A is pointing to the teeth, which are modified scales. They're essentially scales that are formed into sharp structures, grabbing prey and chewing on chunks of meat. Okay, and then there are, count them, 5. 1, 2, 3, 4, 5 gill arches behind there. So this basically represents gill arch number 3 through 7 gill arches. So although I say there's 5 gill arches, what they represent basically is gill arch number 3 through 7, 3, 4, 5, 6, 7 of the lamprey. And so it's homologous. These are all homologous structures. And you can see that here with this specimen. This is the cranium, the mandible, the hyoid, and all 5 gill arches all combined together. I won't have this on a test because it's so kind of delicate. I can't really put labels on it. So on the test, you'll see. The best way to kind of compare it would be like, here is, this is a cranium from like a dogfish shark. This is not the jaw that goes with the skull, so the jaws are kind of small here. But basically, they kind of operate like that. This is a cranium for a shark, and this is a jaw for a shark, not the same shark. But that's kind of how they roll, is like this. They're not attached. And you've probably seen, we can pull it up, like Shark Week on Discovery Channel, when they show like great white shark feeding, they show like their kind of jaws kind of shoot out of their mouth or kind of push out of their mouth. That's because the jaws are actually not physically attached to the cranium. It's just by a ligament. So they can actually kind of move their jaws separately from the cranium. But essentially, jaws were a modified gill. And this is a really cool example of homology where we have exaptation, a new use for an older structure. A respiratory structure has been turned into a chewing jaw. And we're going to see it go further from that. Okay? Okay, so for test questions, if you see some labeled stuff there, be sure you pay attention to that. Let's go ahead and actually I want to jump ahead and talk about the bony fish. So the osteichthyes, the bony fish, we have kind of a piranha here, which would be like a souvenir you could get maybe in South America. And we have a perch skeleton, and then we have some skulls here from a snakefish and a garfish, snakehead and a garfish. So the bony fish, as the name implies, their cartilaginous skeleton has been replaced with bone, so calcified bone. Not only do they have what's called a true skull, and I'll define what a true skull is in a second. Their skeleton is actually a lot more elaborate. The shark's skeleton really just amounts to a backbone and a pectoral and pelvic girdle. So the shark doesn't really have a super complicated skeleton. Here is the shark skeleton. It's kind of an AI model of it. Here's the cranium and jaws. Here's the 5 gill arches. And then there's a pectoral girdle, which is basically a branch off of the backbone. And then a pelvic girdle, a branch off the backbone. But otherwise, there's really no ribs. There's no ribs to speak of in a shark. It's all supported by muscle. You're going to see that when you cut through the side of the shark. You're not cutting through any ribs at all. It's all basically just backbone. Once you get behind the pectoral fins. Whereas with the bony fish, there is a more elaborate skeleton that includes processes off of the vertebral column, which amount to ribs and dorsal processes, etc. So the skeleton basically is a lot more elaborate. But the main difference here with the fishes versus the sharks is all of this is combined. The cranium, the jaws, and the gills are all combined together into what's called a skull. So the definition of a skull is when you have your cranium, your jaws, and your gill arches all bundled together in a big unit. And a lot of that's been replaced by bone. So let me go ahead and see if I can draw a bony fish skull here. Something like a trout or something like that. Okay, this is gonna require a little thought here. So let me show you where the original cranium, mandible, and gill arches are, and you can kind of get an idea of how this works. So the cranium is still there, but it's basically reduced down to just one bone on the front of the head. So this is the remnants of the cranium. It's called the frontal bone. But the cranium is, I guess you call it like a cranial bone. It's no longer a full cranium, it's just a plate on the front of the head. All right, this is the weird part, is, and I'm gonna go ahead and draw in blue what the rest of the skeleton is, the rest of the skull is. The blue is basically all bones that represent, stuff that wasn't in the original shark design here. So we'll do this. Okay, so that's kind of an example. Let me help if I put like an eye on there. So this is all what's called a skull. And a skull is the cranium, the mandible, the hyoid, and the gills all combined together. In the case of a bony fish, the mandible, the mandibular arch, is now reduced down to just the little hinge that goes between the upper maxillary bone and the lower mandibular bone. In a bony fish, for example, the maxillary bone is the upper bone, the upper maxillary is kind of the upper teeth, and the mandibular bone is the lower jaw bone. That little piece of cartilage that cushions the upper maxillary bone and the lower mandibular bone, that little cushion is called the temporomandibular joint. And you've probably heard of that. It is called the TMJ to you. Your temporomandibular joint basically is a little cartilaginous piece that cushions your jaw against your skull. So this is called the TMJ, but really this is the mandibular arch. And that is a piece of cartilage that cushions the jaw as it articulates off of the upper skull. And so the shark mandible now is reduced down to just a little cartilage pad. Some people have TMJ issues, and my suspicion, or just kind of my thoughts on this are, it's ancient cartilage. This is cartilage that goes all the way back to a shark ancestor. And so it's likely some people have osteoarthritis or an autoimmune disorder where your immune system kind of attacks that cartilage because it's different from all the rest of the cartilage in the body. That's the TMJ cartilage. It's that's like your inner shark right there. So if you have pocket jaw.
My body hates the shark inside of me.
The hyoid arch. So here is the TMJ here on the snake head. Is the joint between the maxillary and the mandible. The hyoid arch is this kind of plate right there. This is the upper hyoid bone, or hyoid bone here. And this is the, often called in fishes, the ossicle. And ossicle refers to the hardened bones in your inner ears. You have ossicles also. It's called your stapes incus aemilius. It's the three little inner ear bones that you have in your ears. That's basically your hyoid bone. So in fishes, the hyoid, which was a support arch in the shark, has now basically become the ear bone. This is how fishes hear, or they sense vibration. Then there is a overall plate here, kind of like a big plate that covers all the rest of the gills, and this is called this is called an operculum. operculum is gill arch number 3. So gill arch number 3, which was a respiratory arch in the shark and in the lamprey, now has become kind of a big bony plate that covers all the rest of the gills. And then behind gill arch number 3, I'll kind of draw it this way, is 4, 5, 6, 7. There basically are, this is gills number 4 through 7 gills. So anyway, if you look at our little snakehead here, which is a real primitive bony fish, well, look at that, you can see some labels from the previous test, we'll figure. So label C here is the mandibular arch, which is the TMJ cartilage remnant. Label B is the hyoid arch. Label A is the operculum. And if you look in the back of the fish's head, you can count them 1, 2, 3, 4 gill arches. There's only 4 respiratory arches in this bony fish skull. And so basically what that means is, I mean, I think we're still following this homology. There's a total of 7 different structures. This is number 1. This is number 2. Number 3 is the operculum. And number 4 through 7 are now the respiratory ones. This is number 1, number 2, number 3, 4, 5, 6, 7. You essentially are seeing that the front arches are being modified for other things, right? How cool. So this is technically truly a fish skull because it is the cranial bone in the front, and everything is all combined into one unit as a package. And so really everything's been replaced by bones. The cartilage pieces are basically now remnants and used for other things. Sound good? Okay, so let's go back and I'll talk about some more features of sharks versus bonyfish in a second here. But let's see here. Sharks, they don't have any real buoyancy organ in their body except for their liver. One thing you're going to notice when you cut the shark open is the entire body compartment is full of liver. And the liver is the largest organ in the shark. Presumably because the liver, because it makes oils, helps the shark maintain a little bit of buoyancy. So if a shark stops swimming, they sink because they're negatively buoyant. They're heavier than the surrounding water. But the liver, because it makes so much oil and it's kind of spongy, helps them to maybe not sink so fast. So for a shark, the liver kind of is its sort of buoyancy organ. But the main function of the liver we'll talk about here is it's for detoxifying the food they eat. When they eat food that's kind of rotten or maybe poisonous fish or whatever, the liver basically has a huge food nutrient filtering function. It'll detoxify things that come in food, like food poisoning, etc. So in the shark, they have no separate buoyancy structure like the fishes have. In the bony fish, and we're not going to see this because I don't have a bony fish for the X-ray, but we'll talk about this for lecture exam 4. They have a swim bladder. There is an extension or branch off the gut tract. It's essentially a branch off of the pharynx and esophagus that is an airbag. So the bony fishes have a swim bladder or an airbag, which enabled them to maintain buoyancy in the water. Which is why when you have like an aquarium at home, you can see like goldfish or whatever your ornamental fish that you have, bony fishes, cichlids or whatever, they can seemingly hover in one spot without really moving much. It's because they're using an air bladder in their body to maintain that buoyancy. And for lecture exam 4, we'll talk about the swim bladder and the evolution of lungs. And it's thought that the swim bladder and lungs are probably homologous. But there's kind of an interesting story there that we'll talk about for lecture exam 4. But bony fishes have a swim bladder to maintain buoyancy, whereas the sharks, the cartilaginous fishes, don't. Okay, so we'll kind of come back to that. So I think for the exam, what we're going to do is I'm going to have certainly the shark jaws will be on there and asking you about what those structures are and where they come from. Certainly the bony fish skull, the snakehead skull, and maybe the skeleton. I'm not going to ask you any features of the skeleton, but you should know basically that this is the osteocles, and that all the skeleton's been replaced by bone and cartilage. And then we'll have a shark dissection. So in the shark dissection, we're going to look at kind of some internal organ structures of the shark. And they're very, very similar to what you find in the bony fish, except for the bony fish would have a swim bladder, whereas the shark doesn't. Okay? All right, so let's go ahead and take a look at our shark features here. So for a shark, I'll be going at it. These are dogfish sharks. Their main prey is like soft-bodied mollusks, like probably like sea slugs or really soft-shell crabs, like when crabs are multi-milled crabs. So you can see that the teeth on these dogfish sharks are not really like sharp, slashing teeth. They're more kind of like crushing teeth. So they're kind of feeding on soft crustaceans and maybe some polychaete worms like that. So let's go ahead and I'll just draw some key features of the shark here that we can kind of identify for external body parts. Okay. All right, that looks pretty good. All right, so there's our shark. So some key external features that I want you to look at here, for sure, would be the gill slits. You're going to see that there's 5 gill slits. 1, 2, 3, 4, 5. Not all sharks have 5 gill slits. Some have 6, and there's maybe I think a couple species that might have 7, but 6-gilled sharks are more common. 5-gilled sharks are the most common. 5 gills because the first 2 gills are being used for chewing. But there is a structure above the eye called a spiracle that is a remnant of the second gill slit. This spiracle is used as a sensory structure. They use it to detect speed in the water temperature, water speed. They're using it kind of like an underwater ear, so to speak, to sense what's going on in the water around them. This spiracle is remnants of that second gill arch. It's no longer being used for respiration, it's being used for a sensory feature. So we have our gill arches here. You'll also find that they have nostrils. And so in association with the nostrils, so you'll find these nostrils or nares. So for smelling, sharks have a very, very good sense of smell. And not only do they smell chemical cues, they can also kind of technically smell electrical discharge. So you'll see around the anterior end of the nose of the shark, you'll see what looks like they have kind of like bad complexion. They have all these pores around their nose, and these are basically electro sensors. These are little pits that can sense electricity. So we know that nerve cells basically emit a voltage, right? Like, your nerve cell uses an action potential that goes from -70 to +30. Sharks can smell that. So when there's a prey fish, especially one that's thrashing about because it's injured or such, sharks can actually sense the electrical emissions coming from their prey item. And one way to attract or repel sharks is you can put like a low current DC voltage emitter in water, and at low voltage it will attract them. Of course, high voltage will repel them. But sharks are basically cued into electricity because their prey basically emits that voltage potential that occurs in their nerve cells. Kind of cool. So take a look at those. Take your thumbs, kind of like you'd be squeezing a blackhead or something, and you can kind of squeeze their nose, and this gelatin stuff come out. That gelatin stuff is like electroconductive jelly in their voltage. Obviously, they have the the jaws, etc. Okay, so one other thing I want you to check out here is sharks have what's called a lateral line system. There is a line that kind of goes along the side of the shark here, and this lateral line is a sensory feature that sharks and fishes have. And you can see this if you ever get like salmon steaks or shark steaks or swordfish steaks at the store. What you see in a steak is, if I were to do a cross section between the body here, is you see actually the muscles are organized in a certain way. Let me see if I can draw this here. Okay. So if you've ever gotten like a fish steak, you know it looks something like this. So this is where the guts would be, the intestinal tract, and then this is where the vertebrae is. The vertebrae is. You can kind of see this in the tail of the shark. But they have a. So here's the vertebrae. The tail will be cut in the shark, and you can kind of see this in the tail. But what's going on here is there is a group of dorsal muscles here and a group of ventral muscles, and right between these two, this is that lateral line. The lateral line is full of ciliated cells. These are cells that have cilia in them, so they can sense vibrations. So you probably have seen schools of fishes swimming together, and they seem to kind of like, seamlessly be able to kind of swim without looking at each other. What they're doing is this lateral line is kind of like a pressure sensor, and it has ciliated cells in it. Or if you have an aquarium at home and you've ever done this, knocked on the aquarium, and the fish are like, what? They kind of go crazy. What's happening is the fishes are basically sensing that knock or vibration on their lateral line. So that's how they know something's kind of coming at them to attack them, or what's around them. It's a sensory structure that kind of gives them sort of pressure sense around the whole body. Fast forward, and you know that in your inner ear you have a cochlea that has a coil with ciliated cells in it. That's homologous to the lateral line. So this kind of all moves forward along with the hyoid ossicles. And your ears are basically a combination of lateral line type cells combined with hyoid bones that sense vibrations. So this is all, these vertebrates are really cool because everything's very homologous. No new features, just taking old features and changing them. Okay, and then for fins, I'm not really going to ask these on a test, but we all know this is dorsal fin from shark movies. This is also called the dorsal fin, but it's the posterior dorsal fin. This is the pectoral fin. And these are the pelvic fins. And one thing to note about the pectoral and pelvic fins is bony fishes have pectoral and pelvic fins also. The pectoral fins are gone on this one, the pelvic fins are there. But then we're going to see that amphibians have modified these into legs walking on land. So the homology of fins versus tetrapod, four-legged vertebrates is kind of interesting. And it kind of basically has the same basic structures, at least with the bony fins, not so much with the sharks. But the pelvic fins. Then the tail here, this is what's called a heterocircle tail. And what's interesting here is it's called a heterocircle tail because the backbone kind of goes up into the dorsal side of the tail. So you'll see the shark basically has a tail where the top lobe is larger or more pronounced than the ventral lobe. Whereas in a bony fish, they have what's called a homocircle tail, where the backbone basically stops just short of the tail, and the tail is pretty symmetrical. So that's kind of cool there. I I won't ask that on a test, but you know, that's kind of one of the big key features of sharks versus bony fish is tail structure is kind of a unique difference. But the sharks, the bony fishes are using kind of their bones to support fins, whereas sharks are using cartilage and muscle to support fins. Okay, I think we're moving on here to the shark dissection here. So I don't have an image of this, so I'm going to go ahead and draw it out. I think that'll be it for like all the features I want you to see externally on the shark. Definitely look at the dermal denticles. Remember I was saying that these have these, let's see if I can get a better color here. So the scales are kind of interesting in that they're like little teeth. If you look at them under a microscope or with your cell phone, these are called dermal denticles. And what that literally means is skin teeth. And the actual teeth of a shark are derived from the skin or the scales. So that's kind of a unique feature. Whereas in the bony fish, the teeth are actually derived from the bone of the jaw of the skull. So it's a different origin of the teeth. Okay, so for the shark dissection, these should already have an incision probably between the pectoral fins and probably at the cloaca between the pelvic fins. And what they do is these have been injected with latex. They inject them, I think the arteries will be red, the veins will be blue, and then the hepatic system, the liver system, will probably be yellow. Since COVID, I don't know what's going on, but I think what happened was all of the senior, really, really good biotechnicians that would do this all retired. And now I don't think they really have skilled labor doing this. So in years past, sometimes the sharks are kind of blown up inside. Like they've over-injected them and organs like explode. So I don't know what we're gonna get. So what's happened over the last couple of years is I used to have like all kinds of things to have to know for the sharks because they were really interesting. But in the last couple of years since COVID, the sharks have been junk. So anyway, we're gonna play it by ear here. I think we'll at least be able to get the digestive system and the heart system. If there's some more interesting stuff, I'll point it out. But I think what we'll do is limited to digestive tract and the heart circulation system. Okay. So what we're going to do is you're going to take a scalpel. We have some scalpels over here, and you're going to cut from the cloaca or the pelvic fins all the way up anterior of the pectoral fins. We're going to have to open the whole thing up. Probably would be useful also to do some lateral cuts around the pectoral fin so you can kind of flop it open. Otherwise, you just have a slit and have to kind of pull it open. You want to maybe do some lateral cuts here and here so you can kind of open it like a Bombay doors, you know, like we do or something. So let's go ahead and I will draw what that looks like. You're going to lay the sharks on the back with their ventral side up, and you're going to do kind of a full incision down the belly. All right. All right. Here is our kind of ventral view of the shark here. But it's not quite so chubby, it's just that I kind of flap the body out a little bit. Okay. There's our shark. Here's kind of like the mouth here. Here's the gills. That. Okay, so you're going to do an incision. The little opening here between the pelvic fins is called the cloaca. And a cloaca is a combined genital and digestive tract opening. So it's basically the genital system and digestive tract. There is no external urinary tract on these. Weird. Sharks don't pee. But instead what they do is they retain their urea in their blood to maintain an osmotic blood that will surround the seawater. So sharks basically pee in their own bloodstream in order to get to about 3.5% full osmotic equilibrium. So They're not osmoconformers, they're osmoregulators, but they basically use retention of urea to equal seawater. So if you've ever eaten shark, it kind of tastes a little tangy. It's urea, so they basically taste kind of like urea. So all the tissues are bathed in urea in order to make them isoosmotic. But they do have a separate osmoregulatory organ, which I'll point out on their digestive tract. Okay, so this is kind of like an idealized image. One thing I'll point out here is there is a major kind of division line here. There is this structure here called a transverse septum. And the transverse septum separates, it's a membrane, a septum kind of like the segment horns. It's a membrane that separates the gut cavity, the peritoneal cavity, from the pulmonary cavity, the pericardium, they don't have the pericardium cavity, which is around the heart. So the heart cavities above or anterior of the transverse septum, and the gut structures are all posterior of the transverse septum. So let's focus in on the gut first. First thing you're going to see is the liver kind of fills this whole thing up. So just pull the liver, there's two lobes of the liver. Pull the liver to the side. So I'll draw the gut first, and then I'll show you the liver. So the gut tract of every vertebrate basically has the same parts. So that's kind of cool. There's a big homology in all the vertebrates. The guts all look remarkably similar. Coming through the transverse septum, the esophagus turns into the stomach. The stomach here is on your left side, so it's sharp left. Basically, your stomach is kind of off to the left side of your abdominal cavity. The stomach basically transitions into the intestine at what's called the pyloric valve. The pyloric valve is basically it's a sphincter valve that keeps food in the stomach while hydrochloric acid is breaking it down. And then it lets the food slowly transfer into the intestinal zone. And the transition into the intestine is this structure here called the duodenum. So the duodenum is the British pronunciation. That's how I learned it when I was in college, called the duodenum. But you'll often hear doctors, like American doctors, say duodenum. And it's the same thing. Duodenum, which you can say it with like a fast southern accent, or duodenum, which would be like a British accent. It's the same term. And what it is basically is that transition zone between the stomach and the ileum, which is the absorptive intestine. What happens at the duodenum is the pancreas is there, and the pancreas secretes bicarbonate onto the food, so that the hydrochloric acid in the stomach is neutralized before it gets to your filter. You don't want hydrochloric acid in your filter intestine; it causes burns. So the duodenum, one role of the duodenum is to convert the food from being acidic to being near neutral, little fuzzy acid. I'll draw the pancreas in a second. Then there is a kind of a cigar-shaped structure here called the ileum. The ileum, it might be what you'd call a small intestine in a mammal, but it's the absorptive portion of the intestine. And inside the ileum, there is like a corkscrew-like membrane called a spiral valve. The spiral valve is inside the ileum. So I would encourage you to go ahead and make a longitudinal cut. Basically, as each transfers here, rather than just shooting straight through, it kind of corkscrews like a playground slide or something down through the ileum as it makes its way to the colon. At the very end of this structure here, there is an osmoregulatory gland called a rectal gland. I like to ask this on a test because the rectal gland is actually an osmoregulatory organ that basically regulates salt concentration in the body system. So the rectal gland can resorb or absorb salts, or it can be used to excrete salts. In a saltwater shark, you can imagine what it's doing is excreting salts, because they're kind of excessively salty. So they're trying to get rid of salts. But this is the closest thing to like an osmoregulatory organ that they have. Their kidneys are retaining urea and don't actually connect to the outside. It's kind of weird, like they don't actually urinate to the outside. They retain their urea using their kidneys. I'll show that in a second. Okay, so those are all the digestive structures. The stomach, pyloric valve, the duodenum, that's kind of this S-shaped bend. Ileum, it's called an ileum. And it has a spiral valve inside, and the rectal gland. Those would be possible options for asking on the exam. Good? All right, next we'll look at internal organs. And I think probably the easiest thing to do would be to use maybe like a brown color here. So the liver is the largest internal organ. The liver is going to be like, kind of filling this whole thing up here. The liver is, filling almost the entire gut cavity here. This is the liver. In the middle of the liver, there is a gallbladder. The gallbladder is in the middle of the liver lobes. And what you'll see is the gallbladder hopefully will have some bile in it. And bile, similar to bilirubin, but bile is created or stored in the gallbladder. And what happens is bile is secreted into the duodenum along with enzymes from the pancreas, and that's for emulsifying fats. So the liver's primary job is to make fats for fat storage, detoxify the food the shark eats, and this bile is a kind of like a detergent that when the sharks eat a fatty food, it'll break the fats down into smaller droplets or smaller chunks. So bile is kind of like a detergent for digesting fats or breaking it down into smaller structures. So look at that gallbladder, it's right in the middle of the liver. All right, another organ that kind of is liver-like is the spleen. The spleen will be kind of at the bottom of the stomach or below or behind the stomach, pyloric valve, duodenum kind of junction. The spleen is a blood-producing organ. So the shark blood, which would be nucleated blood, red blood cells, is manufactured in the spleen. What's kind of interesting, we'll see that in the rat, if you have a spleen, you might have heard that your spleen can actually be removed and you won't have any negative health effects. The spleen for us, it's kind of like a beneficial organ. It's still there. When you were an infant, or like when you were a pre-infant, when you were a fetus, your spleen was manufacturing your red blood cells. But what happened was when you were born, blood cell manufacturing transferred to your bone marrow, your red bone marrow, and your spleen is a blood reservoir, a white blood cell filtering organ. Basically, it's a circulatory organ that in every vertebrate except for mammals is for blood synthesis or blood manufacturing. Mammals have kind of lost the use of spleen for blood production and transferred it to the red bone marrow. But the spleen is a circulatory organ for making blood. That's why most vertebrates have nucleated blood, is because the spleen makes blood with nuclei. And in fact, fetal blood in humans is made by the spleen. But when you're born, that stops. Yeah. Okay, and then the other organ that I want you to see here, I'll go ahead and do this in a little different color, is the pancreas. Pancreas, I really like to point that out, because the pancreas is a glandular organ that kind of wraps around the duodenum. And the pancreas is really important because it does so many different functions. It's exocrine, meaning it secretes enzymes and it secretes bicarbonates onto the food. It also regulates blood sugar, so it'll absorb glucose and regulate blood sugar in the body. pancreas has all kinds of cool functions. And now you can kind of get an idea of why it's so difficult. Like if you have pancreatic cancer, get rid of it. It's so hard because the pancreas is not a defined organ. It's a big gland that kind of surrounds your intestine. So take a look at that. It looks like bubblegum wrapped around the body. This is the pancreas.
She was a good color. I did it.
And it's like a glandular gum-like
blue.
structure wrapped around the body. Okay, and then finally, for internal organs, I think we've got most of it. Liver, gallbladder, spleen, pancreas. Let's take a look at the kidneys. And the kidneys are embedded in the back body wall alongside both backbones. The kidneys are a glandular material kind of embedded in the back body wall. If you've ever gone fishing and cleaned the fish, you know, you pull the guts out and then you run your thumb up the backbone of the fish, pushing out those brown glands or brown organ structures, spongy structures beside the backbone. You're basically scraping out the kidneys of the fish. And so the kidneys are kind of a coily gland, and what they do is they connect the cardinal veins coming back from the tail to the dorsal vein that goes back to the heart. So the kidneys basically are a filtering urea from the tail, because most of the metabolism of the shark is in the tail because that's where the swimming power is. So the blood's come back to the kidneys. So see if you can identify those. I'll come around and point it out. It's basically kind of a gland along both sides of the backbone. But it's not like a kidney bean like structure like you think of. We're going to see in the frog. Okay, I think that gives us all of the possible internal organs for the shark here. I'll probably have at least 2 or 3 shark dissections, depending on how I feel on the test. You know, and each one will have maybe 3 or 4 questions added. So one of them will probably be focused on the digestive organs in green. Another one will probably be focused on maybe the body cavity organs, liver, spleen, kidneys, pancreas, gallbladder, etc. So that's all stuff that's going on here in the peritoneal cavity. In the pulmonary, I keep saying pulmonary, the cardiac cavity, the heart cavity, I shouldn't say heart cavity. The heart cavity is up here, so the heart is located here. The shark has a 2-chamber heart. I call it like the toilet tank because it kind of works like the toilet you have at home, where you have a tank in the back that slowly accumulates water so that you can quickly flush it. So the heart is located here, and I'll go ahead and label these parts here, but I want to draw maybe a more elaborate one here, because I'll probably have a station just asking you about heart structures specifically. So the ventricle, there is one ventricle, which is basically the major heart pump. All right, and then these are what are called afferent branches. So these are afferent branchioles. It's high blood pressure that's approaching the gills. When we get back from break, I'll talk about afferent versus efferent. But I always remember afferent is approaching a capillary bed, and efferent is exiting the capillary bed. This is basically delivering blood to the gills, and that's what those afferent branchioles are. Hopefully, you can cut all the way up to where you can see the afferent branchioles, and you can see where it goes in to feed the gills blood. And then behind the ventricle is 2 different chambers. There is an atrium. The atrium is like the upper toilet tank where it accumulates low pressure blood from the body. And then behind this is kind of important to see, is a structure called a sinus venosus. The sinus venosus is basically like the freeway merging system of the return venous system. So like when you drive out 299 towards Reading, you have the Highway 5, 299 overpass, and all those, you know, on ramps and off ramps. That's what the sinus venosus is. This is where the jugular veins, the cardinal veins, the lateral veins, the hepatic vein, all dump back into the sinus venosus. And so it's just basically a bag that collects all of it. The reason why I'm pointing it out is we're going to see this shark has essentially what amounts to a two-chamber design. There is an atrium and a ventricle, just two muscular chambers. When we see the frog, there's going to be a three-chamber design. There's two atria and one ventricle. Well, just to spoil the punchline, the sinus venosus becomes the second atrium, or the right atrium. And so, so the sinus venosus is already there. It's just that the atria kind of orient sideways. This becomes the right atrium, that becomes the left atrium frog. But take a look to see if you can find that. But this is a two-chamber design. You know, I don't think I need to really draw it separately. I can just go ahead. That'd be good enough. So if I had heart questions in the test, we would ask about the ventricle versus the atrium versus sinus venosus. Probably wouldn't ask about the afferent branchioles. But basically, what this heart is doing is pumping deoxygenated blood to the gills so it can pick up oxygen and release CO2 load. And I'm pointing that out because we're going to see that the lung circuit, what frogs have as lungs and what rats have as lungs, basically has to be adapted from what's already there. It's kind of cool how that works. Okay, I think that would probably be pretty good. I'm trying to think of what else. If you're super motivated, if you're super enthusiastic about this, I think it'd be kind of cool to see the shark brain. And I was mentioning today in class that what a brain is a brain is a combination of all your ganglia stuck in one spot in your head, right? So let's take a look real quick. This is an optional dissection, I'll tell you what, right? And I know when I say optional, it means only people who are already getting A's want to do it. But what you do to do the brain is you go into the dorsal side. So here's the dorsal side of the shark, where we have our gills are here like this, and then we have our eyes are like here like this. And what you'll feel is between the eyes, you'll feel like a bump or a dome between the eyes. That's kind of like the top of the cranium, right between the eyes. Anyway, what you're going to do is you're going to take a scalpel, and rather than cutting down like you would like slicing, you're going to kind of whittle away, like you're going to slice like you're whittling a stick. You're shaving off top of the cranium of the shark. And what's going on there, let me see if I can use a different color for the brain here. The brain has 5 lobes. I'll just go ahead and draw the lobes first, I'll kind of describe them. So this is optional, but some people are interested in this kind of stuff.
Bunny ears.
It does.
So the brain kind of looks like this. There's 5 lobes of the brain. So this is all in the cranium. So you're going to have to like shave off not only the top of the skin, but the top of the cartilage of the cranium to actually see all of these. And it's about maybe like 1.5 inches or 2 inches of length. So it takes some doing. But once you open it up, you can kind of get an idea. The first thing you're going to probably see, well, let me just label these and then I'll tell you what you're going to see, is the first lobe here. Let me use blue here for my coloring. The first lobe here is called the telencephalon. The telencephalon is olfactory. That is the smelling lobe, and it goes to the nasal cavity. So there's actually an olfactory nerves that go out to the nasal area. I think it also receives some of those electrostimulus from the sensory pits, electrical pits as well. And you can see that frontal lobe is olfactory. The second lobe is really kind of obscure. It is a It is a glandular lobe that occurs between the telencephalon and the mesencephalon. This is called the diencephalon, second lobe. And this is pituitary pineal. This is the glandular center of the brain, and it's not actually a neural lobe, it's a glandular lobe. So it doesn't look like brain material, it looks like a gland material. So it's kind of brownish rather than whitish. And then some of you saw in the lamprey, that was that third eye that was on top of the lamprey's cranium, was the pituitary gland. But that's a glandular lobe. What that does is it is kind of like light, dark, seasonal cues, sleepy, you know, all those kinds of kind of behavioral gland things. Okay, so then there is the third lobe is what's called the mesencephalon. And this is the optic lobe. This is the one that's really easy to see. It looks just like Barbie butt cheeks. Like if you were to like, you know, if you ever looked at your Barbie, your sister's Barbie, your brother's Barbie, whatever, the little Barbie butt. Everyone's seen it. That looks like, to me, like just like the perfect little butt cheeks of a little doll or something. And watch the rest of the but anyway, it's so striking. You're like, oh my God, it looks like a little butt. And so that is the optic lobe. And what you'll see is that actually has nerves that radiates out to the eyes. There's a really interesting feature. You won't see it unless you go on from the ventral side. But some of those, the right lobe, some of those go to the right eye and some go to the left eye. But there are crossing nerves also, that where the right lobe has a left cross and the right lobe has a left cross. And that's kind of how vision is mixed together. That's how basically when you look out, you don't see two different images. You see a blend, because that mesencephalon is actually combining the image of both eyes into one image. It's actually one image. So that mesencephalon is the optic lobe. This lobe here is the one that's under that dome on top of the head. And what you'll see is it looks like a hot cross bun, if you know what that is. Like a bun or a biscuit that has like a cross on top, kind of like a European thing. But it looks like a kind of like a biscuit or a bun that has like a little T on it or a cross on it. And that's because it has four little lobes on it. This is called the metencephalon. And this is for voluntary motor control. So voluntary motor means that, like, if the shark decides to swim to the surface, or if it sees a prey item that it wants to eat, that metencephalon is what coordinates swimming to go there. So, like, body coordination, swimming, behaviors, it's voluntary motor basically is what the metencephalon does. And then this last lobe is called the myelencephalon. And this is involuntary. This is like respiration, gill respiration, like pulling water in with your pharynx and pushing it over your gills. This is also digestion, your digestive tract, your stomach processes, etc. Your kidney function. So this is like all the involuntary motor type actions that would happen, like if they got something stuck in their throat, like hiccuping or gagging, gag reflex would be myelencephalon. Okay, so what's interesting is this is basically what amounts to the brain. And what we're going to see later on, I'm not going to label it now, but these are all the same lobes you have. Your cerebrum, your thought lobe, is your telencephalon. That's why you don't smell so hot. So your olfactory lobe has been adapted in you to be thought and whatever processing. We do have a pituitary pineal in our midbrain, so that is consistent with us. The optic lobe is present in our brain. We have a cerebellum, which is a mesencephalon, which is for voluntary motor control, and then you have a brainstem, which is for involuntary motor control, like breathing, heartbeat, things like that. So all those lobes are basically lobes we have. It's just that here they're segmented out. You can see they're ganglia. Here we have basically 5 ganglion lobes of the brain, but because they're all combined together, that's why we call it brain and not a ganglia. Got it? So this is optional. I don't have that one checked, but if you want to check it out, it's kind of cool. Sound good? Okay, I know we're pushing time here. You know, that's the way it goes. What I'm going to do here is show you how this goes. I'll get gloves on. And we have these large platter boards here, these kind of cookie sheets. So what you're going to do is grab a cookie sheet for each table, bring it back here. I have my sharks in the bucket back here, and so here's how it goes: is these sharks are probably going to be pretty wet because they're in a bucket of preservative. So what I'm going to try to do is drain as much as I can back into the bucket. But what you want to do is bring your platter up, and I'll plop a shark on your platter, but you may have to drain some of the fluid off of that platter. You can do that in your sinks. Just be sure there's nothing else in the sink. Just whenever you drain off of them, run a lot of water down the sink. This is propylene glycol, is the preservative, which is like the antifreeze in your car. So it's, you know, it's toxic, but it's not toxic to touch. And also, as far as an environmental toxin, it breaks down pretty readily. So that's why I'm saying it's okay to drain the shark a little bit in your sinks, but run it with, like, just leave the water on for maybe 5 minutes after we drain anything out, so it totally goes down. Otherwise, the room will start to smell. Also today is the tabletop might get a little oily. Before you leave, if you could do me a favor and take one of these spray bottles and spray the tabletops down and wipe them off with a paper towel. You can leave them wet, but just wipe them off so we get the oils off. And then also, when we're all done, what we're going to do is we're going to put the sharks into a plastic bag. I'll have a garbage bag back here on this tray. You're going to put your shark and whatever major organs you still have present in the shark. Just go ahead, like if you cut the liver out or cut parts out, or if you cut the stomach open and you find like a little fish or something, leave it in your shark's cavity and just kind of wrap it up. And then we're going to put the sharks in one common bag. All the sharks are going to go in the same bag. If it gets full, I have a separate bag here. But we're going to put the sharks in the bag in the fume hood here so we can get the odors to go out the fume hood for us. So we'll do that. And then your shark boards, of course, will be kind of greasy and slimy. We have industrial soap over there. I'll take all the small trays out of there so we don't get those in the way. But you're gonna ask you a favor is use soapy water to clean these off, and then you see how they're kind of tilted upside down so they drain, and that way we'll have these available for the frogs, etc. We'll use the other sheets for the frogs, but just to keep them clean, keep the odors down so we don't smell up the whole building. That'd be great. Okay. So let's see here. Let me show you how this works. I'm going to go ahead. I don't know exactly what sharks I have in here, but I'm going to go ahead and open this one up and see. And I don't know what sex we have. Males have claspers, and the females don't. This looks like a good shark. There's one in here. So if you guys want to, you can go ahead, Benny, you can bring that chair over. You might need some help there, some support. So, or turn sideways. So like I said, I'm going to try to drain these out. And then this is a male, get a little closer, and then because it has these claspers, so you can show people the claspers. The females won't have the claspers. Okay, so it looks like that bucket's empty. So let me go ahead and I'm going to put this down on the ground, and I'm going to get this bucket, which I think has sharks in it.